It's going to take a little bit of a weird turn as well, so apologies in advance. All the authors up there at the time we did this study were in York, so you'll see references to York, but we've all moved on. York is a beautiful city. Don't let our movements let you persuade or be thought otherwise. So a lot of imaging cerebral physiology and function boils down to how do we understand how the brain works. And kind of this gentleman was my inspiration, and I am going to get you guys involved here. So does anybody know who this person is? Who's my real inspiration for understanding the brain? Anybody?
Ramonica Hall?
No. Pardon? Close. Yeah. Close with Houdini. It is Theodore Animan, who was a mentalist. So you'll have seen Darren Brown, you'll have seen all these guys that can somehow get information out of your brain better than we can as scientists. And as an example of how that works, I'm going to pick on Alex. He's worried about the 46 slides I've got. He's going to be even more worried now. So I could only fit 2 books in my hand luggage on the plane. I've got a sci-fi novel called Sleeping Giants and a mystery called Dark Harbour. I want you to pick one for me. Got to do this quickly, otherwise we're running to come in. Yeah, say that. Like, you're going to pick one.
I like sci-fi.
You like sci-fis, okay. So that leaves me with Dark Harbour. Okay, so in this book, there are about 295 pages. And how many words are on a page in that book? Just quickly, Alex. It's your fault now if we're late for a coffee. 230 words on a page. So what I need you to do, Alex, is look at me. Okay, good. Okay, you're making so many choices. This is brilliant. Okay, so we're going to try and work out how your brain works. I want you to just look on those pages, pick out a random word. The longer the word, the more challenging it is for me. Okay. Okay, so I got the word. Close the book. Yeah. Okay, nice big word. Yeah, you're going to try and challenge it.
It's a big word. Yeah.
Okay. So the 2 ways that a mentalist would do this, they wouldn't use their MRI machines. They would just ask questions and try and probe, right? And they might often tell you to spell the word in your head. So don't spell it out loud, spell it in your head. Okay. Yeah. I want you to think about the first letter. He's smiling, this must be good. Think about the first letter.
Yeah.
Okay. Is it an S?
No.
No. See, this is why I'm a scientist. What letter? What letter were you thinking of?
P.
You were thinking of P. Okay. So clearly that doesn't work. But there are 2 ways you can do this. I said this earlier. Think of the letters. Is this a word that you can visualize? Is this a thing that you can see? Okay, I want you to visualize that in your head. So just like we do MRI, when we fMRI, we're showing a vision into your head. You're looking at that thing. This is something organic. This is something personal. This is a person. This is a person. This is not P for person. This is a person of authority, smiling. Don't give me any more clues. Is it male, female, male, female? Is it a policewoman? Is it a policewoman?
Yeah.
Okay. So, you know, my kind of, these guys inspired me. I went to see Darren Brown. I was like, how does he do it? And why can't I do it with my MRI scanner? And this is as far as we've got, right? We look at it, we try and work out what people are looking, what we're seeing, what they're doing. We stimulate them. We know when we're stimulating, and we use this horrible maths to pluck out these activation maps. And it's all very vague and wrong. And we should all know the problems with bold fMRI in this audience. One being that really when we perform a task, our neurons fire. I drew these myself as well. That needs energy, and we get that from oxyhemoglobin burning with sugar, burning the oxygen in the presence of sugar. That creates deoxyhemoglobin. We get nice blood flow interactions and hemodynamics, but that drives our blood oxygen level dependent or bold signal. To this day, my parents still think I work in washing up liquid. That's only a joke that works in England, where we have a washing up liquid called bold. Back to high school biology. Why on earth do we care about all this? Why does the blood flow change? We've learned a lot about cerebral hemodynamics. We're actually interested in where does the energy come from as physicists, and that comes from the breakdown of ATP to ADP, and we need the oxygen and we need that glucose to restock the ATP through the TCA cycle and glycolysis. As we age, our mitochondria, where the majority of the ATP is made, start to degrade, free radicals There's lots of stuff I've written in the review. I'm not going to repeat it now, it's boring biology. But our mitochondria degrade, and therefore we get less ATP as we age, become less good at producing ATP through ATP synthase. And ATP synthase occurs in the electron transport chain within the mitochondrial membrane. What's important for me as a physicist is that complex 4 and cytochrome have optical absorption peaks at quite nice wavelengths. So 690 here, nanometers, we see that absorption peak for cytochrome. We've got absorption peaks for hemoglobin, which is where you can see your blood when you look at your wrist and it's a different color. And I'll come back to that point later on. And this has been around a very long time, since the 90s, the idea that photobiomodulation, shining red light upon a system, has proved its health. And specifically to the mitochondria is you photo-activate the cytochrome, make it work harder, and therefore as your mitochondrial function drops, you can make the remaining healthy mitochondria work harder and therefore offer yourself some kind of neuroprotection in the levels of ATP that remain. So again, this has been around since the 70s. I talked about this with my mother. It was like we used to do that in the 70s. So she always brings me back down to earth when I talk about my science. However, even since the 70s, the idea that you can shine a red light onto somebody and make them better has been met with skepticism. I myself, when this project landed on my doorstep, was like, that'll never work. I can do the phosphorus spectroscopy, but this will never work. Okay. So we have to remain skeptic about, is it possible to reactivate those mitochondria with red light? How can we measure mitochondrial function with MRI? We can look at phosphorus. Again, another thing from the 70s. We feel like we do these things that are new. They were all in the 70s. That's what I'm learning as I get older. Everything was done in the 70s. So 31p magnetic resonance spectroscopy, you sacrifice that spatial, quite often, dimension to get spectral information. And then you can basically track where ATP, your levels of ATP are from your PI pool and from your PCR pool. In terms of the biochemistry, we have this equation at the top. So we start with ATP in the middle. That will break down into either ADP or inorganic phosphate on the right. And then ATP synthase will bring it back, or creatine kinase will bring it back into ATP. Nice thing about phosphorus spectroscopy and MR is that we can magnetically label the gamma ATP, or the third phosphorus in ATP. So we just put a load of RF into it, saturate it, and then when it breaks off the ATP and heads into the phosphocreatine or into the PI pool, we will also see a drop in those signals. Okay. The longer we leave that for, the more of my label would have made it back. These are reversible reactions, so some of the label can come back into my original pool. And of course, there is a T1 effect as well. But that's all known. I didn't invent any of this. We just irradiated it and tried it, looking at photobiomodulation. So hit the gamma ATP, and then you can see in the red line there, there's a drop in the PCR signal and the PI signal as well. You can do lots of control experiments to prove that it's not the application of your magnetization at gamma. It's very long pulse, and you can put it somewhere else and double check the attenuation elsewhere. But yeah, my label ends up in those pools and I see a drop in those signal. The longer I leave for these reactions to happen, the more signal drop I essentially get. So yeah, this is the maths behind it, and the data on the left here. Again, this is at 3 Tesla, so it's quite noisy. We can zoom in on the PI intracellular and extracellular peaks. We're only interested in intracellular for mitochondrial function. And fit those to distributions and then subtract off the baseline, subtract off the extracellular PI, and just get my PI intracellular as a function of the time I've left that reaction to occur for, which you can see is from 0.5 to 7.4 seconds. I get an exponential decay, which I can then fit based on some T1 assumptions to estimate the flux rate for that chemical exchange. This is the methods. Again, University of York, beautiful city. We all left. Didn't look back. I mean, yes, did. Oh, terrible mistake. This is actually the scanner in Manchester. I made the film there. But we scan people before. We do a baseline 31p magnetization transfer experiment. We then shine this ridiculous red light on the back of the head for 20 minutes every day for 4 days, and we bring them back in. I had the very same expression that you guys had. I was like, rubbish, rubbish. So there we are, coming back out, and you'll see the bulb in a second. And even when you see the bulb, you'll probably think, God, this guy. So there he comes out. It's time for the photobiomodulation. We train people how to apply this, and they literally sit there with a red light bulb on the back of their head for 20 minutes. It's a hard training, but we managed to get through it. So this is the pre and post data for that exponential decay. Again, saturation time referring to the time I wait from my pulse, RF pulse on gamma ATP to transfer into the other parts of that chemical equation. T2 decay lines, fit them, account for the changes in T1s, and this is the kind of data you get at. So pre and post, there is a difference in the rate of exchange for ATP after the photobiomodulation. We see an increase in ATP synthase, and that is across all the 7 participants we ended up with. This was over COVID. It was very disrupted, and the mean there as well. A few of the participants had to be taken out. Generally older people we were doing this in. You asked people to stay still in the scanner for the 50 minutes this took, and they just couldn't. So we did have to eliminate some of the data. The complete control is to then look at the creatine kinase, which shouldn't be involved in mitochondrial function and should not be excited by the red light. And sure enough, the creatine kinase pathway remains exactly the same pre and post. So I was like, that's kind of cool, interesting. Nice data. It's now published in Aging Cell. Yeah, so become a biochemist, apparently. That's what I'm becoming. We can look at this and we see this increase of PI to ATP in the mitochondria after shining the red light on people's heads when they're old. Now for something completely different. What time am I on? I've got 2 minutes to do this. Okay, so my background, for those that don't know, is actually preclinical imaging, optical imaging. Coming back, how did I get into this? We shine lights on rats' heads and we look at their cerebral hemodynamics. I do that while they're inside the scanner. We can map between cerebral hemodynamics and the bulb fMRI signal to build forward biophysical models of activity. You get blood volume, you get deoxyhemoglobin, oxyhemoglobin from that data. How does it work? Again, back to absorption. We shine light on a rat brain. We put a filter in the way, and the amount of absorption of that light will be dependent on the concentration of oxy and deoxyhemoglobin. They're absorbing at different rates. In the green example, oxy is absorbing more than deoxy. I get the measurement. And then I stick another filter in, and in this example, deoxy is absorbing more light than oxyhemoglobin. I have 2 measurements, 2 equations to work out 2 unknowns, which are my concentrations of deoxy and oxyhemoglobin. Slightly more complicated than that, if you know the Baer-Lambert law for optical imaging, is that you also need an estimate of the path length of the light. For intrinsic optical imaging in the visible, it's exactly the same as photochemistry. Shine a light on through a filter. Normally the test tube width is the lead path length, and then you record that. A little bit more complicated in brain. It doesn't go all the way through, otherwise it would be see-through, and that would be a little bit gross. What we're relying on is remitted light. So we have to do very, very clever Monte Carlo simulations of tracking where light goes, what it bounces off, what it's absorbed by. I can go through that in the break. But essentially, we're looking at refraction and absorption. For optical imaging, we're interested in the refraction, how much light comes out of the brain and therefore can inform me about deoxyhemoglobin. For the phosphorus study, I was interested in the absorption, how much is actually absorbed by the cytochrome. So I'm going to skip over these. We did this. We took the 3D MRI data, putting it down into a very basic 5-layer tissue model of scalp, skull, CSF, grey matter and white matter. Filled that with physiologically real light properties and looked at the light transport. So this was, the same people that went under the phosphorus scanning had a structural scan. We could then work out all the light that was absorbed, and strangely enough, as we can see by this figure, most of the light is absorbed by the skin and the skull, right? Hence why we're not see-through, hence why you can generally only see skin. But if you zoom in on the grey matter, we still get a lot of light that actually makes it down, and those that are in infrared spectroscopy go sigh of relief. That between 400 and 1,000 nanometers, we still get a lot of light into the grey matter. And we can work out from these models that approximately 0.15% of my light at the required wavelength is actually making it into cytochrome. And that's driving this ATP change, supposedly. The other interesting thing we could do, and I'm yet to publish this, nobody wants it, is actually use that model to account for things like skin melanin content, how that would affect it. So the same problem we have with pulse oximetry. Here we can actually say how much longer would you need to leave the light on if you had a dark skin tone. We can look at CSF as you get old, brain shrinks, there's more CSF. I can now account for that. Skull thickness, do we get thicker as we get older? Does it get thinner? We can account for that. So this model can actually really help us. The physical model of light transport can help us in placing the therapy to amplify those ATP changes. So take-home message just before coffee is that we did this first study. This is published in Aging Cell, the phosphorus data. It's the light modeling that isn't published. And we shine this red light on called photobiomodulation or low-level light therapy. And we in older people, we see this increase in ATP flux pre and post TBM, and it's a quantifiable rate that we can give in per second. Then I moved to Manchester. We do a lot of this in sports, so we're now looking at fatigue and muscle damage, less interesting to this audience. So I'll skip over that. Long COVID is also what we're looking at, and we're going to be using Vaso to look at that, and phosphorus to see if there's any change in this. Skip over. These are the people that really make this work happen. I just do magic tricks. So yeah, Elisa's in the audience here. Everybody else has moved on, and Jamie and Jim are at my current institute. Well, that's it. Any questions?